In this video, we'll do an exam review on humoral immunity, which is a part of the adaptive immunity. Few important questions before we do the topic. Question number 1. Which cells produce immunoglobulins? Number 2. What's the role of T lymphocytes or thymocytes in humoral immunity? Number 3. What are different types of B lymphocytes? And number 4, what's the function of variable and constant regions of the immunoglobulin? Adaptive immune system is characterized by antigen-specific response to pathogen. So it's an antigen-specific response to pathogen. Following the initial contact with the pathogen, the subsequent immune response is quick. Why? Because the body's defense cells have memory for that pathogen and recognize it very quick when it attacks later. So which cell bring about the specific immunity against the pathogen? B and T cells, except the NK cells, bring specificity and immune memory to the host defense. The two types of adaptive immunity are cellular immunity and humoral immunity. So let's discuss humoral immunity. Humoral immunity is done by B lymphocytes. B lymphocytes are produced and matured in the bone marrow and reside in the lymphoid organs, and they constitute 10 to 15% of the peripheral blood lymphocytes and 50% of the splenic lymphocytes. So humoral immunity done by B cell, which are produced and mature in the bone marrow and resides in the lymphoid organ and constitute 10 to 15% of the peripheral blood lymphocytes. Humoral immunity is the major defense against the bacterial infection, whereas the cellular immunity is a major defense against viral and fungal infection. B cell produce antibodies after interaction with antigen. So what happens after interaction with antigen? When antigen binds to B lymphocytes, they divide and transform into the plasma cells. Plasma cells have intensely staining Golgi apparatus, and then they produce antibodies called immunoglobulins. So humoral immunity is done by antibodies produced by plasma cells, which are formed from the B lymphocytes. After interaction with antigen, immunoglobulin recognizes antigen by their heavy and light chain variable regions and kill the bacteria, for example strep pneumonia and H. influenza. The antibody binds specifically to the antigen and kills the pathogen, foreign bodies and the toxins. So what's the role of T lymphocytes or thymocytes in humoral immunity? The helper T cells facilitate it, and suppressor T cell inhibit this process. And the activated T cell produced by the thymus regulates suppressor and helper T cells. So the helper T cell facilitate and suppressor T cell inhibit, and activated T cell control the process. Now the types of B cells. The two types of B lymphocytes, number 1 antigen dependent and number 2 antigen independent. The antigen dependent B lymphocytes mature in the secondary lymphoid organs, including lymph nodes, spleen and gut hair patches, and they interact with the antigen and convert it to plasma cell, and immunoglobulin formation occur. The antigen dependent B cell also induce B memory cells, whereas antigen-independent B lymphocytes develop and mature in the primary lymphoid organs. So antigen-dependent B lymphocytes, they mature in the secondary lymphoid organs, and antigen-independent B lymphocytes, they mature in the primary lymphoid organ. The B cell features immunoglobulins on the cell surface have receptors for Fc region of the immunoglobulin G, IgG, and activated complement. They have APC for antigen processing. It has B cell receptor BCR for antigen and secretes specific antibodies. Each B cell one single specific antibody. The B cell has two light and two heavy chains, one active and one inactive chain. This is known as allelic exclusion. Immunoglobulins bind to specific antigens, toxins, bacteria, viruses, and foreign bodies, and each immunoglobulin has two long heavy chains and two shorter light chains. Each immunoglobulin has two domains, a variable region and a constant region. Variable shown in red color and a constant in blue, and the variable region of the heavy and light chain or long and short chain have FAB or the antigen binding sites, and they have a constant region. So they have a variable region and a constant region. The constant region binds to complement surface Fc receptors of the neutrophils, eosinophils, macrophages, dendritic cells, NK cells, and B cells. The light chains are of two types, kappa and lambda, and have one variable and one constant unit. This is a variable unit and this is the constant unit. The heavy chains are of five types and they have one variable and 3 to 4 constant units. So the heavy chains have one variable and 3 to 4 constant regions, and they are made up of homologous sequences and share the same primary structure. Now what's the function of variable and constant region? The variable regions, they constitute antibody binding or sites. And what's idiotype? Idiotype is the specific site in the variable portion that exactly binds the antigen. Whereas a heavy chain determines the type of immunoglobulin. Constant regions are involved in biological functions of the immunoglobulin. The immunoglobulin type depends on the type of heavy chain. Now different types of heavy chains are IgG, IgA, IgM, IgD, and IgE produced by plasma cells, and they are joined together by a disulfide linkage. These are the disulfide linkages. IgG, IgD, and IgE are monomers. IgA is a dimer and IgM is a pentamer. So three monomers, IgG, IgD, and IgE, and IgA is a dimer and IgM is a pentamer. Now let's discuss different immunoglobulins. First of all, immunoglobulin G, IgG. IgG are the most common immunoglobulins and they constitute 75 to 85% of the immunoglobulins. They are the lightest immunoglobulins, are monomer and have four types. Deficiency of IgG increases the susceptibility to bacterial infection. It crosses the placenta because it's very small and may present at birth. The Fc portion of IgG binds to the neutrophils, macrophages, large lymphocytes, and activates complement. So immunoglobulin G are the most common and the lightest ones and constitute 75 to 85% of immunoglobulins, and their deficiency causes bacterial infection. It crosses the placenta and it binds to the different cells and activate complement. Now IgM, immunoglobulin M is a pentamer and heaviest immunoglobulin. It is the first immunoglobulin that appears in primary immune response, and it is also the first immunoglobulin to appear in neonates. But the presence of IgM at birth indicates congenital infection, for example cytomegaloviral infection. IgM is important in autoimmune diseases, for example IgM against IgG of the rheumatoid factor in rheumatoid arthritis, collagen diseases, some infectious diseases like subacute bacterial endocarditis. So immunoglobulin M is a pentamer and is the first that appears in primary immune response and is the first immunoglobulin to appear in neonates. Its presence at birth indicates congenital infection, is also important in autoimmune diseases like in rheumatoid arthritis and collagen vascular diseases. Now IgA, immunoglobulin A is a predominant immunoglobulin in secretions, for example in tears, saliva, in GI and respiratory secretions. Two types of IgA. IgA1 primarily in the serum and IgA2 is primarily found in the secretion. It's the one which is dimer. It fixes complement via alternate pathway and has potent antiviral activity. So immunoglobulin A is a dimer and is predominant immunoglobulins in the secretion. Two types, IgA1 found in serum and IgA2 is more in the secretions and it has more antiviral activity. What's the mechanism of IgA function? In the intestine, bacterial and viral antigens are transferred to the lymphoid tissues and stimulate lymphoblasts. Lymphoblasts then enter the circulation and mature there and then move to lymphoid tissues, intestine, lungs, breast, genitourinary tract mucosa, and secretes IgA. Now IgE binds to mast cells and basophils via Fc receptors, and the interaction causes the release of mediators of immediate hypersensitivity. Now answers to the questions. Number 1. Which cell produced immunoglobulins? B lymphocyte divide and transform into plasma cell, and the plasma cell has intensely staining Golgi apparatus and produce antibody called immunoglobulin. Number 2. What's the role of T lymphocyte or thymocytes in humoral immunity? The helper T cells facilitate it, and suppressor T cells inhibit humoral immunity, and activated T cells produced by thymus regulate these suppressor and helper T cells. Question number 3. What are different types of B lymphocytes? The two types. Number 1, antigen dependent, and number 2, antigen independent. Antigen dependent B cells mature in secondary lymphoid organs including lymph node, spleen, and gut hair patches. Antigen dependent B cells interact with antigen and convert it to plasma cell, and immunoglobulin formation occurs. They also induce memory B cells, whereas antigen independent B cells, they develop in the primary lymphoid organs. Question number 4. What's the function of variable and constant regions? Variable region constitutes antibody binding FAB site, and a heavy chain determines the type of immunoglobulin. Constant regions are involved in biological function of immunoglobulins.